You see, medical parasitology isn't some isolated subject. Think of it more like the central hub of a wheel connecting all these different disciplines. To really truly understand a parasite, we have to study its morphology, what it actually looks like. We need to know its complete life cycle and its method of transmission. All that knowledge then feeds directly into both laboratory diagnosis and the much broader field of epidemiology, which is how we track these diseases as they spread through entire populations. So here is the formal definition that really ties it all together. Medical parasitology is the complete science of these organisms. It covers everything, from the initial study and identification, all the way to the active prevention and control of the diseases they cause out in the real world. All right, now that we've defined the field, let's meet the primary subjects of our study. We're going to break them down into 3 major classifications so we can really understand the kinds of threats they pose. First up, we've got the protozoa. Now, these are microscopic, single-celled organisms. And here's the crucial point to remember. They multiply inside the host. This can lead to incredibly rapid, overwhelming infections. Some key examples you'll see are Entamoeba, which is a type of amoeba, Giardia, which uses that little tail-like flagellum to get around, and of course Plasmodium, the sporozoan that's responsible for malaria. Our second group is the helminths, or what we more commonly call multicellular worms. Now, unlike the protozoa, these are large parasites. You can often see them with the naked eye in their adult stages. They generally fall into 3 main subgroups. You've got the nematodes, which are roundworms, the cestodes, which you probably know as tapeworms, and the trematodes, commonly called flukes. And finally, we have the arthropods, and these guys play a really fascinating dual role. On one hand, you have vectors, like a mosquito. Now, they don't cause the disease themselves, but they act as biological taxis, transmitting it from one host to another. On the other hand, you have ectoparasites, like mites, which cause a direct infestation right on the skin. In both cases, they're the bridge between the environment and the human host. Knowing the parasites is really only half the story. Now we need to understand their intricate dance with the hosts they depend on. And this is a seriously complex relationship, one that's often been shaped by millions of years of coevolution. This table lays out all the different roles so clearly. So on the host side, a definitive host is where the parasite hits sexual maturity. An intermediate host, well, that's where the larval or asexual stages happen. A paratenic host is just a transport vehicle. No development happens there. And a reservoir host is what maintains the parasite out in nature, allowing it to stick around. Then, on the parasite side, some are obligate. They absolutely must have a host to survive. Others are facultative, meaning they can live freely. And they can also be permanent, living their whole life in a host, or temporary, just visiting for a short time. And that brings us to life cycles. A direct life cycle is pretty simple. The parasite completes its whole life in just one host, like the Ascaris roundworm. But an indirect life cycle is way more complex. It requires one or more intermediate hosts, just like we see with Plasmodium and malaria. Understanding this difference is absolutely critical because it tells us exactly where we need to intervene to break that chain of transmission. So how do these parasites actually breach our defenses? This infographic shows all the different routes they can take. It could be the fecal-oral route from contaminated food or water. It could be vector-borne from the bite of an insect, direct skin penetration from parasites lurking in the soil, or even congenital transmission from mother to child. Each route of entry presents a totally unique challenge for prevention. Okay, we've covered a lot of the theory. Now we're going to make a crucial pivot to practice. Let's see how we apply all this knowledge to stay safe in the laboratory. Because let's face it, understanding a threat is the first and most important step to managing the risk. The laboratory environment has multiple hazards. Of course, there are the obvious biological ones from the infectious agents themselves, but we also face physical risks, like needle sticks, procedural risks, like inhaling aerosols while you're handling something, and chemical exposure from reagents. And I want you to notice that red alert box. It lists some of the very agents we've just been talking about, like Entamoeba and Plasmodium. This just highlights that the stakes are incredibly high. And that brings us to the specific protocols designed to protect us. Look, these aren't just suggestions or guidelines. In a high-risk environment, they are your first, your last, and your absolute best line of defense. It all starts with personal protective equipment, or PPE. Every single piece has a purpose. A buttoned lab coat protects your clothes and skin. Protective eye gear shields your eyes from splashes. A face mask prevents you from inhaling aerosols. Disposable gloves provide that crucial barrier for your hands. And closed-toe shoes protects your feet. Every piece is non-negotiable. Beyond the gear itself, we operate with a mindset called universal precautions. The core principle is simple, but it's absolute. You must treat every single sample as if it is infectious. That means no eating or drinking in the lab, ever. Mandatory hand washing, and constant disinfection of your work surfaces. It's all about building a culture of safety into every single action you take. Safety is also about process. When a specimen arrives, there's a strict checklist. First, you check the patient ID. Then you verify the container is appropriate and totally intact. You check for any leakage, and only then do you register its entry. Each one of these steps is a checkpoint for both safety and for the integrity of the data. And when the work is done, you can't just throw things away. Safe disposal is critical. Autoclaving uses pressurized steam to sterilize waste. Incineration gives you complete destruction of pathological materials. And biohazard bags make sure that contaminated items are safely contained until they can be disposed of for good. This is how we prevent infections from ever leaving the lab. Finally, let's talk about documentation. I know it might not seem as immediate as PPE, but accurate records are just as vital. Keeping meticulous logs of specimens, results, quality control, any incidents, it all ensures complete traceability. If something does go wrong, these records aren't just a history. They're the key to understanding how, when, and where the process failed, which is how we prevent it from ever happening again. And now, let's bring it all together. We're going to connect the microscopic world of the parasite with the disciplined, practical world of the laboratory professional and see how knowledge and safety are truly, well, two sides of the same coin. This scale just perfectly captures the core message of this entire explainer. On one side, you have knowledge, understanding the classifications of parasites, their life cycles, and how they're transmitted. And on the other side, you have safety, the unwavering discipline of universal precautions, sticking to your PPE, and proper disposal. The thing is, you simply cannot be effective in this field if one side outweighs the other. They have to be held in perfect balance, always.